Yes.
Dr. Perry, still on the topic of the PFC's relationship with other bodies, I wanted to ask you next about the relationships with BPL. First of all, on a personal level, we've heard evidence about the relationship between Mr. Watt and Dr. Lane. Do you have any particular recollections of the relationship between Dr. Cash and Dr. Lane?
No, not a great deal. I think there was some. They had conflicting views, which I think probably arose from the discussions that took place in the 1970s and perhaps early 80s about how the overall supply of UK plasma should be fractionated in Scotland versus at BPL. But I attended meetings where they were both present, and there was always a constructive dialogue. I don't think they agreed on all issues, but as I said about Mr. Watt, they were both strong personalities and had clear views on certain topics.
And then we've heard evidence that at an informal level, and I have in mind the written evidence of Dr. Smith and then the written and oral evidence of Dr. Foster and Dr. Snape, that there were good informal links and communications between, for example, those working on research and development in PFC and those working on research and development in BPL. What was the nature of your own interactions with BPL? Who did you tend to have dealings with?
I had dealings with Dr. Snape, Dr. Smith, Dr. Smith's co-worker, Dr. Winkleman, Dr. Lane, although probably more frequently with Dr. Smith and Dr. Snape and Dr. Lane, but I had fairly regular, though not frequent, contact with Dr. Lane, and of course I met him periodically at meetings, and we would always have constructive discussions, I think.
Now, if we just go to your statement to this inquiry, so WITN 6920001. Yeah, WITN 6920001. And if we go to page 15, bottom half of the page, you say in paragraph 46, sorry, actually, let me pick it up in paragraph 45. So you say there that PFC considered its relationship with BPL to be very important, although as separate units under different UK jurisdictions, there were few, if any, mechanisms or fora for regular formal liaison and collaboration. However, there was a regular and productive scientific collaboration. And then if we go to the next paragraph, please, Sally. You reiterate in the opening sentence of paragraph 46, you don't recall any formal liaison mechanisms between BPL and PFC, although Professor Cash was a consistent and strong advocate for closer and more formal cooperation between UK blood services concerning the development of safer NHS products. And then, if we look at the last sentence on that page, you say, I'm not aware of or cannot recall any formal mechanisms being established by DHSS, SHHD or the blood services for formal cooperation concerning joint project development programmes. And then if we just go over the page, you then set out, I don't need to go to any particular paragraph, but you talk about your own relationships with BPL personnel, and communications in particular between Dr. Foster and Dr. Smith. If we go to the next page then, please, Sally. And just look at paragraph 52. You say there may have been some merit in a joint approach for the development, production and supply of plasma products for the UK-wide NHS, particularly for providing increased benefit of scale for PFC, but this did not apparently enjoy the support of DHSS or SHHD to the extent of serious consideration or study. Can you just assist us, first of all, with what you mean by the passage in brackets, referring to providing increased benefit of scale for PFC?
I think it was simply that, that the PFC, as it became set up for Scotland and Northern Ireland, was a relatively small fractionator in international terms. There were, certainly compared to BPL, and certainly compared to some of the larger units in France and the Netherlands and so on. And as with most endeavours of this type, increased scale creates greater efficiency. So I think PFC always felt, certainly during the 1970s and 1980s, that it would benefit not only PFC but the UK as a whole to have a more equitable split of plasma to supply to both PFC and BPL. But by the time I'd arrived, I think that that discussion, I don't think it was concluded or it was over, but it was certainly well advanced to the stage where it seemed to me that Scotland would pursue its programme of self-sufficiency for Scotland, and with the prospect of Northern Ireland coming on stream as well.
You said in one of your Penrose statements, I'm not going to put it up on screen, but the reference is PRSE 0001258, page 11. But you've said in that, or you observed, that it would be difficult to argue against a case for the closest possible collaboration between two UK NHS services engaged in identical and unique activities. But as the statement that we've got on screen explains, your sense appears to be that there wasn't the support of either the Scottish Home and Health Department or the Department of Health in London for formal, closer, collaborative endeavours.
No, I think that was my sense when I joined the PFC, that these discussions have been going on some time, and I think there was the shift experiment that was done to still establish whether or not PFC could increase its throughput and so on. But I have actually thought about this, and the first point I'd make is I think the arrangements that we had, the informal arrangements as they're called, were highly productive. And it may well have been in the context of haemophilia product development that a more formal arrangement for collaborative working between BPL and PFL and PFC may not have improved the outcome. For example, and I'm sure we'll come on to heat treatment. I think I think at BPL and PFL under Dr. Jim Smith's leadership, they chose to go down a dry heat treatment route, and we, in the early stages of our heat treatment program, wanted to do pasteurization because we thought that was the best technology to adopt in terms of non-A, non-B hepatitis. I think it's possible to speculate that had there been a formal arrangement, then it could well have been that both organizations might have chosen to simply pursue pasteurization, because that was, I think Dr. Jim Smith has mentioned this in his witness statement, that was, I think there was quite a strong consensus between PFL, BPL and PFC that was the preferred technology if the target virus to be inactivated was non-A, non-B hepatitis. So it may well have been that in a more formal, structured arrangement, those involved in the discussions of the technology to choose may well have chosen pasteurization. In which case, I think the outcomes in terms of the speed with which both BPL, PFL and PFC developed their dry heat treated products would have been much slower.
If we leave aside the research and development element, and I understand your answer in that regard, but look at what the benefits might have been of a more formal liaison in other terms. A more formal liaison or arrangement between Scotland and England and Wales and Northern Ireland might have resulted, might it not, in increased overall production of domestic concentrates?
Yes, I'm not absolutely clear in my mind whether the constraint on BPL's output was capacity or plasma supply, or perhaps a combination of both. But I think PFC certainly took the view that it did have spare capacity, or spare capacity could have been developed fairly quickly with additional resources, freeze dryers and so on. But the central processing technology had the capacity to produce more product. So yes, I think is the answer to that question.
And did you ever get any sense of what underpinned the lack of enthusiasm on the part of SHHD and DHSS that you described for a closer joint collaborative exercise?
No, I don't.
Can I move then to relationships with fractionators beyond BPL and PFL? First of all, to what extent did PFC have links with fractionation centres outside of the United Kingdom?
We had, well, I think Mr. Watt, who set up the PFC and commissioned it and so on, he made it his business to appraise himself of the work of many fractionators. These were predominantly not-for-profit organisations in Europe, but they were commercial organisations that he had dialogue with as well. So that extended into my tenure as well, and we had really quite excellent relationships with the Dutch Red Cross, the Finnish Red Cross, I think latterly the French fractionation facilities under the CRTS as it was then. So yes, quite an extensive network of contacts, predominantly in the not-for-profit sector.
And in terms of the for-profit sector, the commercial fractionators, the pharmaceutical companies producing their factorates and so on, to what extent was there any relationship, formal or informal, between PFC and those pharmaceutical companies?
I think we knew who the commercial companies were, and I think we knew some of the key, particularly scientific staff involved in those activities. I don't recall there ever being a free exchange of information. I think, as so many others have said, the commercial sector kept their processes and their know-how very closely guarded secrets for obvious commercial reasons. So although we would have dialogue, we had a broad general understanding of what they're doing, but I don't think we ever accessed at any deep level scientific understanding of their processes and how they made them work.
It might be said, having regard to Dr. Foster's evidence, that a picture emerged of being able to glean bits and pieces here and there at international conferences on small pieces of a jigsaw.
Yes.
It would presumably, in principle, have been enormously helpful if there had been a more extensive or formal process for the exchange of information.
Kind of global cooperation. Well,
Yes,
yes, but I think for reasons that I've described, they were. The not-for-profit sector was much more like a family in Europe. They weren't competing. The national services in France, Finland, Holland, the UK weren't competing with each other for markets. So there was a much freer exchange of scientific information and cooperation. The commercial sector were fiercely competitive with each other and still are. So they guarded their proprietary information and know-how, as I say, very, very closely. They were closely kept secrets. So there was never, in my view, any real prospect of accessing their information. And if one did, then more often than not it would be patented. So not accessible for the public sector to utilise.
Now, moving then back to England and Wales, but now the regional transfusion services in England and Wales. Is it right to understand that the PFC, you didn't have much by way of dealings or interactions with the regional transfusion directors in England and Wales?
I think that's a fair summary, yes. I think we knew many of the regional directors and they knew us, but these were informal contacts met at conferences and meetings and
Did you ever, for example, get the minutes of the meetings of the regional transfusion directors of England and Wales?
I think I probably did see them, yes.
Would that have been through Professor Cash?
That would have been through Professor Cash.
Can I then turn to relationships with haemophilia centres and haemophilia centre directors? We've obviously touched on that in relation to Scotland because of your earlier evidence about Professor Cash's principle of everything going through the regional transfusion centres. Did you have a closer relationship with degree of interaction with the haemophilia centre in Edinburgh for reasons of geographical proximity than other services, or was there no real difference in Scotland?
I think it's probably true to say that we did have a slightly closer relationship with the centre in Edinburgh. It was adjacent to the transfusion service in Edinburgh, and it was literally 5 miles from the PFC. And Professor Ludlam was the chair of the Coagulation Factor Working Party. So yes, we did have a close working relationship, but that wasn't to the exclusion of other haemophilia directors.
In relation to the haemophilia centres in England and Wales, I'll come on to Northern Ireland in a moment, but in relation to England and Wales, there was no particular tie, or as I understand it, or regular degree of interaction, other than through a degree of PFC attendance at the annual meetings of the UK Haemophilia Centre directors.
I think that's correct. I think that's true.
And I think we see you attending the UK HCDO meeting fairly early on in your PFC career, October 1981, and you were there again, I think, in 82 and 83.
Yes, yes.
What was the thinking behind having PFC represented at those meetings?
I think my Mr. Watt, who I reported to, felt it was a good idea that I should be exposed to these meetings because in the absence of a formal training program, this was a very good way of learning what the issues were and what the priorities were and what was happening in haemophilia care. So these were very important meetings. I don't think I probably contributed greatly to them, but I certainly listened carefully and learnt a great deal at a personal level. But they were UK meetings, so they were there to represent the Scottish haemophilia directors and the Northern Ireland haemophilia directors as well.
And then specifically in relation to Northern Ireland, you had contact with Dr. Maurice McClelland as the regional transfusion director through his attendance at regional transfusion director, at SMBTS meetings once the arrangement with Northern Ireland was established. What about the haemophilia centre in Northern Ireland and the haemophilia centre director, Dr. Main? To what extent were there any interactions between the PFC and Dr. Main or her centre?
I never went to the haemophilia centre in Belfast, but I think as soon as PFC started processing Northern Irish plasma and supplying them coagulation factors, albumin and immunoglobulin products back, Elizabeth Main, I think it's correct that she joined the annual meeting of Scottish Haemophilia Centre directors and transfusion directors, but she certainly attended the Coagulation Factor Working Party on a regular basis. So we had quite a close relationship with Dr. Main. She was a good colleague.
Can I then come on to some aspects of regulation? We've obviously touched on the issue relating to the manufacturer's licence. Just in relation to that, is it right to understand that Dr. Cash and Mr. Watt, neither of them agreed with the decision not to renew the manufacturer's licence?
This would have been prior to 1984.
Yes.
I think, as I've said previously, I think both Mr. Watt and Professor Cash felt that formal regulatory approval of PFC was an important objective. But I think certainly following the inspection by Flint and Purvis in 79-80, I think there was probably a view taken by both Professor Cash and Mr. Watt that it would unlikely to an application for a manufacturer's license would have been unlikely to have been granted at that stage. So the absence of the manufacturing license became an important, I guess I would call it an important tool to try and get the funding that we felt we needed.
And just for the benefit of others in the transcript, there's a reference to the views of Mr. Watt and Dr. Cash and their disagreement with the decision not to renew the manufacturer's licence in a document put together by Mr. McIntosh for the Penrose inquiry. I'm not going to go to it, given that it predates, I think, your involvement. PRSE 302556, page 16 is the reference for that. But before I ask you a little about the process in relation to product licences, I just want to ask you about interactions with another regulatory body, NIBSC, so the National Institute for Biological Standards and Controls, if I've got that right. What dealings and interactions did PFC have with NIBSC?
We had I guess in some senses, a formal relationship with NIBSC as the National Control Authority, whose responsibility, amongst many other things, was to approve batches for release, I think, latterly in our relationship, to do virological testing, to test product batches for potency and so on. So that was the formal relationship, and it was very close and mirrored very much other organisations that had products on the market in the UK. But there was also a slight, I guess you would call it a more informal relationship between scientists in PFC and the wider SMBTS with scientists at NIBSC. For example, Dr. Cuthbertson was quite closely involved with NIBSC in developing assay techniques for testing for virological markers in plasma products and adapting test systems that were available and designed for single donation use to assay systems that would be sufficiently sensitive for testing plasma products. So that's an example of the sort of collaboration. And I think NIBSC also set up various working parties. I think I've seen documents that have been sent to me, and I certainly recall them of an AIDS scientific working party that was established by NIBSC. So NIBSC, I think, part of its vision, certainly in the early 1980s, by its then director Geoffrey Shield, was to become much more integrated in the work of the transfusion service, and the best way to do that was to set up working groups on scientific issues.
Can we just look at your statement to this inquiry, WYTN 6920001, page 13, please, Sally. I just wanted to ask you a little more about paragraph 40. You described the PFC being required to submit samples of finished products, intermediates and plasma pools for control testing and batch release to NIBSC as the National Control Laboratory. Can you just help us understand a little more about how that process worked? So what samples would be submitted to NIBSC, and at what point or what stage in the life cycle of a product would that take place?
Okay, the arrangements, I can certainly explain what it meant. What I can't give you is a precise timescale for the various changes that took place. But in the very early stages of this, I think samples of PFC product would be submitted to NIBSC for factor VIII potency testing, for example, to check that the potency that we were assigning to the product matched their determination. And that was important because finding assays that were suitable for plasma products was not always easy. So there was an ongoing collaboration in that area. I think as time went on, there was a requirement for following the introduction of HIV testing and probably hepatitis B testing. We were required, or we did. Perhaps we weren't required because we were operating under Crown immunity, but we took the view that we should engage and integrate our activities with in common with the commercial sector and anyone else that's putting products on the market in the UK. So the NIBSC, as I say, would determine whether or not our assay systems, so we would send a sample of a batch of factor VIII, for example, and we'd already tested it, and we found it negative for HIV and negative for hepatitis B surface antigen. That product, that finished product vial would go to NIBSC and they would test it and confirm the result. And I think when this system operated, we wouldn't have released the product until we had confirmation from NIBSC that they too had found the product to be negative in their assay systems. And then it eventually, I think it further developed into a phase of requiring plasma pool testing. So we would take a sample of the plasma pool. Actually, strictly speaking, it was the cryosupernatant, not the plasma pool. It was the cryosupernatant, and that would be tested by PFC for virological markers. And a frozen sample would be sent to NIBSC, and they would test it for HIV, hepatitis B surface antigen, and eventually hepatitis C in the early 1990s when that test was introduced. And similarly, the pool would be released, although because of the nature of the process, we couldn't delay the processing of the pool until we got the result back from NIBSC, because the turnaround time was sometimes a fortnight. But the pool would have to have been found negative for the finished product batch associated with the pool to be released for use. And the reference to intermediates, what's that a reference to, samples of intermediates? It could well have been that at some stages, intermediate fractions like fraction 2, this would have perhaps been, or maybe, well, it talks about plasma pool, so it might have been part of a more informal collaboration on a scientific level. I don't, on reflection, I don't recall there being any specific regulatory requirement for submitting intermediate fractions for testing by NIBSC, but we may well have done that as part of a scientific collaboration. So in terms of the contribution of NIBSC to virological safety, its ability to contribute really depended upon the availability of screening tests, or tests for markers of a virus. Primarily virological markers, but it also had this important role of standardizing factor VIII assays, and NIBSC, amongst its many other things that it did create international standards for factor VIII, and it was always a matter of concern when a new international standard was coming out, because at a stroke, a change in the international standard could have reduced the measured yield in PFC products, and this problem would have applied to BPL as well by as much as 20%. So they had this very important role of creating factor VIII reference standards against which operational routine standards would be calibrated. And then can we look at a letter that you wrote in late 1987? It was referred to in one of the extracts from your Penrose evidence that we looked at before the break. It's at PRSE 0000712, please, Sally. So this is a letter you wrote 21st of December 1987 to Mr. Donald, the General Manager at the Common Services Agency. It's headed Product Liability / Personal Liability. You say, picking it up in the second sentence, I note the agency has not and perhaps cannot provide an absolute assurance that it will underwrite the activities of its employees at the Protein Fractionation Centre. I'm also aware that professions such as pharmacists and doctors can arrange personal indemnity insurance. Unfortunately, the professions represented at PFC, including my own, do not have access to such facilities, and thus I must conclude that in the face of existing laws of negligence and the new consumer protection laws, I and my senior colleagues remain exposed and vulnerable in an area of product manufacture which attracts considerable public attention. And then just continuing with the next paragraph, perhaps my sustained anxieties stem mainly from the fact that the manufacturing activities of the centre are at a level substantially above that for which the centre was designed,
and there exist major breaches of GMP in our day-to-day activities as a consequence which can only be resolved by the provision of additional buildings. One might argue, therefore, that I'm professionally negligent in allowing such activities to continue. However, I continue to do so in the interest of self-sufficiency and, in my opinion, in public interest. The agency is aware of this position, but to my knowledge has never explicitly instructed me to continue with a policy of growth. I'm therefore knowingly operating the centre above its capacity, and I'm ever conscious of recent disasters elsewhere whereby the corporate body and individuals are subject to criminal proceedings. As the captain of this particular ship, I find the analogy too close for comfort. And then over the page, you conclude, clearly I'm not implying an imminent disaster at PFC, since we continually make strident efforts to compensate for deficiencies at the centre, but I believe it is appropriate and reassuring for myself and senior staff that we clarify unequivocally that the agency is aware of the deficiencies at the centre and has authorised the continuation of activity at the present level. I recognise that this amounts to an instruction to carry out our professional duties out with minimum standards required of the pharmaceutical industry, but in the circumstances such an instruction will at least relieve some of the anxiety felt by myself and senior colleagues. Now that doesn't read like a letter that would be written lightly, Dr. Perry.
No, I do remember this letter. Yes.
What were the particular concerns that drove you to write in those terms in 1987 to the CSA?
I think the background to this particular letter was an increasing level of frustration that we were not getting approval or agreement by SHHD or the Common Services Agency for that matter, although they would have got the money from SHHD for what was required at PFC, which was a fairly major building program involving cold rooms, storage areas, increased and improved processing areas, new sterile filtration areas, and so on. So this was really, I think, part of a process. And I think you showed the letter from Professor Cash. This slightly predated that, and Professor Cash was responding to this letter. I think this was my attempt to do pretty much what it says in the letter, which is if I am, and I was committed at the time. I didn't write this letter with any prospect of closing PFC down. That wasn't my intention, but it was to try and raise personal, and I think they were legitimate concerns at the time that I was responsible, I would have been considered responsible, and I didn't have recourse to things like medical defence unions or the associated body for pharmacists to protect me in the event that there was some litigation that was directly attributable to the circumstances. So I was actually asking for the CSA to understand the issues and underwrite the activities of PFC, and also to try and force funding to be accrued. Yes.
And did you get a response from the CSA at any stage?
I think there was a response. I don't think it was a satisfactory response, because that's what provoked Professor Cash to write his letter, and the response was from the SHHD. I don't think the CSA really understood what the issue was, because they very, very seldom engaged in details such as this in their activities. But SHHD, I think, responded by saying, in our view, the PFC is perfectly suitable for its purpose. And I think that's what promoted Professor Cash to write his letter, which in some senses was underpinning and emphasising the points that I was making. But he, in his typical way, he went a little bit further.
We can take that down, thank you, Sunny. I want to ask you next then a little about product licences, specifically focusing on, obviously, Factor 8, Factor 9 products. First of all, at the PFC, whose job was it to make the applications for the product licence or have any ongoing interaction with the medicines division, with the licensing authority?
I think that was probably assigned to me when I joined in 1981. I think that was one of the portfolio of activities, and I think, as I briefly mentioned before, one of the major tasks that I undertook in 1983 was to assemble a comprehensive product license application, which was an enormous undertaking at the time, for our newly developed intravenous immunoglobulin product.
And what did you understand the purpose of the product licensing system to be?
It was, I guess, effectively a peer review system that we felt that self-regulation, particularly in the area of complex biological manufacture, we felt it was professionally appropriate that we should submit our studies, our processes, and their intricate designs to peer review. And the only peer review system that existed was product licensing. And that, for example, was the rationale I made for applying for a product license for intravenous immunoglobulin in 1983. I did get a response when the SHHD discovered that I had submitted this license to the MCA. I think the chief pharmacist expressed his concern that I had, I think in his words, usurped the authority of the Secretary of State.
And I'm just going to ask you to look at part of your evidence to the Penrose inquiry on the licensing system in broad terms, PRSE 0006025. So this is an extract from your evidence, well, this is the transcript, sorry, of your evidence on the 13th of May 2011. If we go to page 42, please. We can pick it up, I think, at line 16. This is in a question that was being asked to you. It says, what you're referring to there is that the licensing of products, I think the idea in your statement, please correct me if I'm wrong, is that the fact that a commercial product was licensed would inform the doctor or the clinician as to whether or not it was appropriate to use that material. In other words, if it was licensed, then it was all right to use it. Is that what you were suggesting? And then we have your answer. I think that's what the licensing system is intended to achieve. It's intended to give prescribing doctors, top of the next page, clear indications that the product is safe, it's efficacious, its risk/benefit balance has been properly and objectively assessed, and it is suitable for use absolutely. And then there's a question, sorry, if we go back to the whole page, there's a question or an observation put to you along the lines that the evidence the Penrose inquiry had heard was that licence didn't make any particular impact upon the clinical decision-making of haemophilia centre directors. And then if we go to the bottom of the page, you were asked if you wanted to comment on that, and at line 21 you said only to say that at that particular time I think the licensing and the continued licensing of products was part of the confused world that we operated in. I think, as we discussed previously, the notion of removing licences for these products on the basis of what we now describe as a precautionary principle would have created so much disruption in the treatment of patients that it was considered an inappropriate thing to do. Sorry, can we have the rest of the first half of the page, Solly? Because it's not, in fact, that point I want to ask you about at this stage. It's the next bit. You continue, I would still take the view that the licensing system was and is set up to establish that products can and should still be used safely in clinical use. That doesn't preclude an individual doctor for a whole number of reasons not using a particular product, but I agree there is a slight conflict there. The system, you know, as we now know, was not as effective as it might have been, but the consequence of creating a safer environment was to expose patients to no treatment at all, certainly with concentrates. Now, I'm showing you that for present purposes, really just to understand what you thought the purpose of the product licensing system was. It was intended, is this right, to provide a degree of external assessment of the safety of the product.
Yes, before you showed me that, I was going to clarify that that was the other major reason, and prescribing doctors, although they understood the status of PFC, they had a reasonable expectation to understand what measures we had taken to assure the safety of these products. And the only system that exists for doing that is the licensing system.
And so if we then turn to the question of the product licenses for factor concentrates at the PFC, we'll see probably this afternoon when we look at some of the examples of package inserts and product warnings, we'll see that license applications were submitted in, I think, 1978,
Yes.
signed by Mr. Watt,
Yes.
for product licenses for Factor 8 and Factor 9, so for NY and for DFIX. So that was, I think, in 1978, and they would therefore have come up for renewal in around 1983.
Yes, that's right.
As far as you can recall, were those licenses renewed in 1983 for the factor concentrates?
I think they were, but I would have to check on individual products and so on. But I believe there was an attempt made to, or perhaps a successful attempt to get them relicensed and their licenses extended.
Now,
but I would add that the original license application in 1978, although it was written carefully, it probably wasn't as substantive as the type of license application that would have been required by a commercial manufacturer to submit. But the key information was included, and the licensing authority took the view that it should be licensed.
now, during the lifetime of a product license, so in relation to PFC's product license, if we to say from 1978 to 1983, at least in relation to the first license, were there any particular reporting obligations or reporting practices undertaken by the PFC in terms of, for example, adverse reactions, incidences of hepatitis, jaundice?
Yes.
Would those in the life of the licence be reported to the licensing authority, or would they be reported elsewhere? Do you know?
Certainly latterly it was quite carefully prescribed when the licences became substantive following the removal of Crown immunity. Any significant adverse reactions, and there would be a definition what significant meant, they certainly would be reported or in regular post-marketing surveillance studies and so on, not necessarily on a one-by-one basis, but they would be collected together and submitted to the licensing authority. I think in the early 1980s, it was certainly the case that regional transfusion centres were encouraged to have a close liaison with the users of the products, and if there were any adverse reactions, they would be reported directly to PFC. So I think we had a fairly good idea. I wouldn't call it a fully developed pharmacovigilance system as one witnesses nowadays, but it was certainly a link between individual patients. I think interestingly though, it may have been the case that factor 8 infusions to patients so frequently caused elevations of ALTs and perhaps even jaundice. I don't know the clinical details, but that was prescribed as an expected adverse effect. So that wouldn't necessarily have to be recorded. So we didn't get routine reports. This is before introduction of heat treatment of patients seroconverting for non-A, non-B, for example. But we would have got reports of a hepatitis B transmission.
And if you'd had this may be a hypothetical question because I don't know if it happened, but if you got reports of a hepatitis B transmission reported to you at PFC, do you know whether in the period really leading up to the mid-80s, or indeed beyond that, the period when there was Crown immunity, did that get reported by PFC or by SNBTS more generally to the licensing authority?
I can't be sure of that. I can't be sure of that. It would certainly have been reported to the PFC. PFC would have carried out an investigation and the appropriate outcome implemented, which would have ranged from no action to a complete recall of the batch.
And then can you recall, and if you can't, obviously please say so, but can you recall whether information on pool sizes was part of the information that PFC would submit to the licensing authority when applying for a license?
I think it was, actually, yes. I think there would have been a requirement to state it was a measure of the scale of activity, which was an important parameter, I think. I don't know whether it was expressed in kilos, litres, or individual donations, but one or all of those would have been used to measure it. I think that's the case, yes.
And then, again, during the lifetime of the licence, you've got your licence granted for 5 years. You don't have to renew for 5 years. During that period of time, if there are changes that might have a bearing on risk, for example, increase in pool sizes, or the development of knowledge that non-A, non-B hepatitis is a more serious condition than hitherto believed, or the development of knowledge that non-A, non-B hepatitis is, or the belief that it's an inevitable consequence of a first dose with NHS concentrates. That kind of information that might have a bearing on risk comes to light in the course of the duration of a product licence. Do you know whether there was any obligation on the licence holders to notify the licensing authority? Or was that just something that would be picked up at the next renewal?
I think the licensing authority were interested in adverse events which fell outside the recognised adverse events, which would have been things like slight hemolysis or even transmission of hepatitis viruses. The licensing authority would have regarded those as fairly frequent and not remarkable. We now know that they were extremely remarkable, but at the time, it was as we now know, a significant and severe problem with the products. I do recall one example of a factor VIII infusion taking place, for example, in Glasgow, and the patient developed a significant reaction to his treatment, and that was reported and thoroughly investigated. And I believe actually a medicines inspector came up to look at that. It turned out that it was a result of a too faster infusion by the patient. I think it was finally determined to be an issue of citrate toxicity rather than a problem with the product batch. But that was reported to the licensing authority because it was unusual, and I think we hadn't seen it before, so we felt it was appropriate to report it. But as I say, the pharmacovigilance systems that we have now are much more significant and robust.
Yes, and just so that you understand, Dr. Perry, the purpose of these questions is really just to try and get a sense of how in practice the licensing system, the product licensing system operated. We've looked in earlier hearings at some of the legal requirements. We've looked at some of the applications from commercial companies, but it's really the extent to which there was once the licence was granted, the extent to which there was an ongoing dialogue with the licensing authority at this period, late 70s, first half of the 80s, that prompts the questions.
I think it wasn't fully developed either because the licensing authority. I talked in my Penrose. I think you showed it as in the Penrose, and it was a confused area. And I think it was confused not only for PFC and the SMBTS, but it was confused for the licensing authority as well, that they weren't quite sure how to deal with an organisation that was submitting product licence applications when actually they operated under Crown immunity. So the status of that relationship was a little bit confused.
Do you think, if we take the example of increased pool sizes, do you think it's more likely than not that wouldn't have been reported by PFC to the licensing authority during the lifetime of the licence? Or are you just not able to say?
I think it would have been an unusual event, but it's difficult to define what a small change and a large change might be, but a small change in pool size would have been considered not of consequence, of sufficient consequence to notify the licensing authority, because they would simply note the position, bearing in mind that most of the products that they were licensing which were sensitive to pool size were commercial concentrates with pool sizes orders of magnitude larger than the PFC. So if the PFC had submitted a license application or an amendment to a license application saying we've changed we've modified the pool size from 500 litres to 1,000 litres, I don't think they would have taken much notice of that. I think they would have said, well, that's still orders of magnitude less than the size of, or substantially less than the size of for example, commercial pools.
And then the example of developing knowledge in relation to non-A, non-B hepatitis. Would it be right to understand from your earlier answers, and please correct me if this is not right, that a growing awareness of the seriousness of non-A, non-B hepatitis wouldn't be something that PFC would specifically report to the licensing authority because post-transfusion hepatitis was already a known risk?
No, I think we would have assumed, or I would have assumed, that this was a well-known phenomena by the licensing authority, and they didn't need reminding from one manufacturer that that was the case.
Can I then just ask you a little about the PFC's involvement, or I think more accurately, lack of involvement in issues relating to donor selection? If we pick it up again in one of your Penrose statements, PRSE 0001823, please, Sally. And if we go, I think, to page 3. No, that's not. Oh, yes, it is. I think, yes, sorry, second paragraph. No, top of the page, please. Thank you, Sally. So you say in the second paragraph, latterly during this period, PFC and regional centres worked more closely on the development of quality systems and standard operating procedures for the processing and testing of plasma. And then this, but this did not extend to issues of donor selection, which at that time would have been accepted as the exclusive responsibility of regional directors and their medical staff. This situation remained largely unchanged until reorganisations of the service in the 1990s. In its original licence applications to DHSS Medicines Division for Factor 8 information on donor for Factor 8, information on donor selection practice or policy was neither supplied by PFC/SNBTS or requested by the UK Licensing Authority. So, as I understand it, 2 points essentially emerging from that part of your statement. First of all, in submitting your licence applications, you were neither asked to provide nor volunteered information about donor selection practices in SNBTS.
I think my understanding is that's the case, yes.
And then secondly, your broader point is that the approach to donor selection was not something that PFC got involved with. It regarded that as the term used here, the exclusive responsibility of the regional transfusion services.
I think that's correct as well. That subsequently changed as the quality systems developed, and PFC was also, I was personally instrumental in trying to expand the concept of quality systems throughout the regional transfusion centres, driven by the need for PFC to have a modern and mature quality system in place. But I think it was still the case that the detailed considerations for donor selection were, I think personally, and I think generally, were considered to be the exclusive responsibility of regional transfusion centres. That's not to say that I was excluded from an interest in that, but certainly in the early 1980s, that would have been a given, that the plasma coming to PFC had been subject to suitable donor exclusion and testing procedures.
May I ask, on whose behalf was the application made for what would in other what was originally and would after the mid-80s possibly have been had you been a commercial organisation? Who made the application? SNBTS?
No,
or PFC?
It was, well, neither actually. So it was made, I think, and I'm not sure when this changed, but maybe never, it was made on behalf of the Committee of Management of the Common Services Agency.
And they managed both SNGTS and, the regional transfusion.
They were the
centres
umbrella organisation that managed the SNGTS and many other organisations.
This was one organisation which was responsible, albeit vicariously, for donor selection, as it was responsible for the information that you were capable of giving it. So why wouldn't the information about donor selection have been given to the licensing authority?
I think it probably would nowadays, but I think if the period that we're talking about is the early 80s or the late 1970s, it is the case that the licensing authority didn't seek this information. Nor, incidentally, I imagine, did they seek the specific information of those commercial suppliers of Factor VIII in terms of their donor selection procedures and so on. So it didn't have the focus of attention which we now know it should have done.
Yes, thank you.
And we can just see the first page of a licence application. If we go to PRSE 0002726. Just to confirm that if we go to page 5, I think, Sonny, confirm the answer that Dr. Perry just gave. Oh, no, that's not it. Sorry, I've got an unpaginated version. What I have doesn't resemble that at all. Can you go on 5 pages, Sonny? Yeah, there we are. So if we look at, if we zoom in on the top half of the page, we can see this is the Factor VIII product licence application, and then we can see the proposed licence holder is the Committee of Management, Scottish Health Service Common Services Agency. Right. And then trading style to be shown on licence if different from above, Scottish National Blood Transfusion Service Protein Fractionation Centre. And then if we go just to the bottom of the page, we can see that this was the licence application submitted by Mr. Watt in March 1978.
Yeah, on behalf of the Management Committee,
yes. The Management Committee. We'll come back to that document for different purposes at a later stage, Dr. Perry. Just sticking, however, with the issue of donor selection just for a moment, do you recall whether the PFC ever did its own audit or assessment or checks on the donor selection policies and practices that were being implemented by the different transfusion centres in Scotland?
I don't think it would have considered itself competent to go into the detail of donor selection procedures, but it would have checked and audited against the need to have donor selection. Standard operating procedures for donor selection would have been the sort of working title. So the PFC would have taken the view that it wasn't competent to judge specifically what the exclusion criteria might be, but they would. But we would check to make sure there was a documented system in place and that was operated. Perhaps not certainly not in the 1970s or the early 80s, but that developed over time. The whole concept of PFC auditing regional transfusion centres as a supplier of a raw material. I can't remember the date on which in which it emerged or developed, but it was sometime in the 1980s, I believe.
Now, I want to move to the topic of self-sufficiency next and get started on that before we break for lunch. If we look at PRSE 0006011, and again, this is an extract from the oral evidence given to the Penrose inquiry, 24th of March 2011. And I want to go to page 94. We looked at one passage on this page earlier. I want to look at a different passage now. So you were being asked here about your arrival at and your role at the centre. I just want to pick it up at line 11 onwards. You say, I think the preoccupation at that time, and there was absolutely no doubt in my mind that this was the case when I joined in 1981, and certainly strengthened as the 1980s moved forward, that the dominant goal and target was self-sufficiency. It was very clearly evident to everyone who worked in it that in terms of plasma products, the goal and the aim was to make Scotland self-sufficient in plasma products and in particular coagulation factors. Does that remain, first of all, your recollection of the position?
Yes, a very, very clear recollection, and that became very clear to me very soon after I started in 1981. It didn't take long for me to understand why the PFC was there.
And do you recall, if we keep this up on screen for a moment, because we're going to go to a different page in a moment, do you recall gleaning or gaining an understanding of why self-sufficiency was this dominant goal for the PFC?
Yes, I think the complexity of the answer to that developed over time, but initially it was very clear to me that there was a view that plasma derived from voluntary non-remunerated donors and used as a raw material for plasma products was a lower risk material than plasma or products obtained by the commercial sector. So the object of self-sufficiency was to effectively eliminate the need for commercial products to be used in Scotland. That was the stated aim. It was quite clear. It was unambiguous and clearly, and I believe everyone that I came into contact with understood that.
The reason for asking Dr. Perry is that there might be a number of reasons why it said self-sufficiency is a good goal. It might be, for example, the elimination of commercial concentrates, because ultimately that will be cheaper for the state. So that's why I wanted to pick up upon your understanding of the reasons for it. If we go on in this transcript to page 118, picking it up at line 5, you come back to your understanding about this being a goal. My understanding at the time when I joined the service as a new person to the blood service was that this was a goal or a policy that had been set by the Scottish Executive at the time, that we wanted to meet the WHO recommendations for self-sufficiency. And then you say this, but I think also it became very clear that one of the prime justifications for self-sufficiency was a belief, which was based on fairly good evidence, that imported products from the USA, which were the alternative source of products, were much higher risk products than those that would be produced from voluntary, non-remunerated blood donors from one's own community. So it was a target which was aimed at creating a sufficiency of supply from our own community, but also a target which sought to reduce the risk to haemophilia patients of transmission of disease from other countries. So that remains your evidence that does it, that one of the prime motivating factors, or was it the prime motivating factor, was the issue in relation to safety?
I think it was the prime motivating factor. I don't believe there was ever a comparison of what the cost savings is. It certainly wasn't driven by a perceived lower cost. I'm not sure that it was lower cost to produce one's own products rather than buy commercial products. So I think that was the clear justification, and it was a goal set well before my arrival in SNBTS by SHHD, who I don't think there's any formal grand statement by SHHD that we will become self-sufficient, but it was a concept that they clearly supported and funded, because collecting increased levels of plasma required significant injection of revenue funding for blood bags and the consumables and so on associated with this.
And then in terms of your understanding of what self-sufficiency meant, I think it's probably in this same document at page 57, please, Sally. No, it's not. Sorry, wrong reference. Can we go to PRSE 0006025? PRSE 0006025, and then page 57 of that document. So this is your evidence to the Penrose inquiry on the 13th of May 2011. We don't need to go to it, but at the bottom of the previous page, you've been asked what was the goal that you took yourself to be working towards in terms of self-sufficiency. And then this is your answer. Meeting most, if not all, the needs for plasma products in Scotland, with the exception of occasional rare products, individual patients who had idiosyncratic reactions to the product that we had on offer. We would fully accept that it would certainly be justified to use a non-NHS product. Do I understand that last sentence to mean justified to use a non-NHS product in the exceptional circumstance that you described in the previous sentence?
If an individual patient had an acute reaction or a severe reaction to our product, and the haemophilia, if it was a haemophilia product, and the haemophilia director judged that his or her patient needed an alternative product, then I'm not sure that the PFC or the SMBTS would have had any involvement in that. If that's what the haemophilia director in Scotland decided was the case, they would have purchased it. And I think periodically that did happen.
But leaving aside what you describe as an exceptional category of cases,
yeah.
You understood self-sufficiency for Scotland as meaning the production of sufficient factor concentrates to provide for treatment at a level to be determined by the haemophilia centre directors for all haemophilia patients in Scotland.
Yes, I think that's correct. There were some very, very rare coagulation deficient patients, factor 13 deficient, or maybe factor 5 deficient. And I don't think PFC ever had an aspiration to make every product for every patient. So we had, as we've talked earlier about the productive relationship we had with BPL, that I think from PFL, Dr. Smith had available products, factor 13 products, factor 10 perhaps, and if we needed those products, or if haemophilia directors needed those products, they could readily obtain them from BPL.
And so this would encompass producing and therefore providing enough factor concentrates, at least in terms of factor 8 and factor 9, for home treatment needs for patients? For all the estimated needs, and I think part of the SMBTS self-sufficiency planning or work up to it was work that was carried out by Professor Cash and other senior haematologists in Scotland in the 70s and 80s, and they sat down and they identified in some considerable detail how much factor 8 would be needed for the haemophilia population of Scotland. And the figure that sticks in my mind, I think it started out lower than this, but the figure that they arrived at was 2.75 million units per million population. And I think if one reads the minutes of the annual meeting of haemophilia directors and transfusion directors, you will see that every year that figure, that aspirational figure, or that target figure, was underlined and agreed to by haemophilia directors, by SHHD and others attending the meeting. So it wasn't just a blind aspiration to be self-sufficient. There were numbers placed on this. So I've got a number of other questions relating to self-sufficiency, but perhaps we can pick those up after the lunch break.
Yes, well, let's do that then. We'll come back at 2 o'clock.
Thank you.
2 o'clock, please.